Okay, welcome to the Intro to Algorithms Part 4. This should be the section right after you've solved the finding frequent words problem. So we're going to pick up on finding frequent words this time, except moving into a case where we don't know where that origin of replication is. And so how do you define what is a frequent word in a DNA sequence? Before we get there, let's over here. So you'll have to download the Intro to Algorithms for PowerPoint. And there's one problem here, number 15, that I got out of order. Doesn't really matter. You've had to solve this one already in some ways. So you should have code that you can kind of repurpose for this if you've already solved number 11, regardless of which way you've solved it, actually, whether you did it the pattern count way or the dictionary way or the frequency array method. They all required you to slide along the string, and then, well, maybe I take that back. Maybe if you did it the dictionary way, you didn't have to exactly have this code. But in any case, when you're sliding along and you get your window, and in this case, like in the sample data for number 15, ATAT, on the first check, right, you say if ATAT equals text in that window, it's false, so you wouldn't count that as a match. But in the second window, it does match. And so the only difference for this problem is that rather than add one to a value in a dictionary or in the list of frequent windows, you just have to record the position. So what is the position of that window where you got the match? Well, it's just the I in the current for loop. If you're using I as your index, right? For I in range length of text minus k plus 1, right? That for loop. So when you check ATAT against the second time through the loop, I will equal 1. And so that's what Rosalind is looking for. It will match again when it gets to position 3, and so on. So really, in this case, instead of doing the counting, which is actually a little bit of a harder problem, here, this was like an intermediate problem where you just needed to be able to output the start position of the windows that match. All right, so hopefully not too bad on that one. You should already have code that can do that. Just modifying it a little bit to get the right output. So how do we find frequent words? Well, if we go back to our Vibrio cholera origin of replication, right? That's our Vibrio there. We had a 500 base pair region where we counted the most frequent words. Well, what happens if you actually do that is that we have ties. And so which one of these hidden messages constitutes which one of these frequent words or frequent k-mers constitutes the hidden message. And so that's where we get into sort of some interesting questions about the probability. So how likely is it that you would find any particular 9-mer in this case appearing 3 or more times in a sequence? Or you could sort of flip the question a little differently. We'll see later. They're just going to give us the value of the it needs to occur t times or more. But what is that t, right? How often does it appear before we start to get interested? Actually, do this. Get a little bigger. Right, so there's two kind of probabilistic models that can allow us to get at that question. One is called the multinomial model. These are the simplest set of models where the fundamental assumption is what I have underlined there. It's just that the nucleotides, these same kind of models can be used for amino acid sequence, but since we're just talking about DNA, I'll just stick to DNA sequence. These multinomial models just assume that every position is independent of all the other positions. And so the most simple basic model is just to say, if each nucleotide appears at equal frequency, so they all are frequency 0.25, then the probability of seeing one of them is also 0.25. And so then you just, what's the probability of getting an AC as my pattern, or an AT-Kamer? Well, it's just 0.25 times 0.25. And so that's what this little equation is, right? You're just multiplying the frequency of each nucleotide by 0.25. And again, I say that's the simplest one because, a little bit slightly more sophisticated one would be, okay, what's the actual frequency of those nucleotides in my DNA sequence, right? So if I have, for example, a GC biased, meaning more than 50% of the nucleotides are G or C, I could increase the frequency of these as long as all of this has to sum to 1, right? Because all the nucleotides have to sum to 100% to account for everything. So if I make C 30%, for example, something else is going to have to go down by 5%. Right? And so that might give you a little bit more appropriate estimate of the likelihood that you're going to see a particular sequence. And think about it in the extreme, right? If you had a stretch of sequence that was all A's, it would be silly to think of the probability of calculating that as 0.25 times 0.25 right? You can't get C, G, or T in that sequence. It's all A's. So, right, you would want to model that as the probability of A being 1. Right? You aren't going to find any variation. So, on one hand, not an interesting case, but it also demonstrates, what's going on with this likelihood estimation, probability estimation. Okay. The other class of models are Markov models, and these are a little bit more interesting because you can model more biological reality in some sense. What I have illustrated here is just a first-order Markov model where all it does is, no matter where you are in the sequence, it estimates the probability of going from one nucleotide to another. So in sort of graph form, it looks like this thing over here on the left. So if the current position that I'm looking at is an A, what's the probability of staying A for the next one? So you would have a sequence that is AA. Or what's the probability of the next nucleotide being a C? That's the probability of AC. And so, right, if you expand this graph out to a table, all of these things in the middle are called transition probabilities. So what's the transition from A to A or A to C and so on? Okay. And then if you sum the probabilities down these columns, for example, you would have what's the likelihood of starting at any one of these sites somewhere in there. And then just another way of looking at exactly the same thing. How would you actually count the probability of a sequence? It's essentially what's the probability of the current nucleotide given the likelihood of the one that preceded it. Okay. And there are lots of ways. So if you end up working in this field at all, where you're trying to predict motifs or really these models extend to trying to predict anything in a genome. Lots of the gene prediction software, for example, you sequence a new genome, you want to know what genes are in there. gene prediction software is based on this too. And so you'll hear of something called a training data set. The training is giving the algorithm something that you already know. So for example, give it the human genome, train the Markov model so that it knows for a human genome what is the transition. Like every time there's an A, what's the probability? Like how often does an A stay an A for the next position? And you can calculate that across the genome to make this transition probability. So you know for a human genome, what's the likelihood of moving from any nucleotide to any other nucleotide as you scan along the genome. And the cool thing is for these Markov models is you can make this sort of arbitrarily complex. You can define separate transition matrices and then have a likelihood of moving from one transition matrix to another. For example, we know that first and second positions are more constrained because they are restricted to, encoding regions, I should say, right? You don't get the same transition probabilities as if you're in a non-coding region, because the amino acid translation table is not random, right? Whereas if you move into a non-coding region, you have, with a bunch of caveats, more or less constrained rates of mutation from one to another. And so you can build in, say you're trying to predict genes, okay, what does a genome where we know what the genes look like? So the human genome, for example, really well annotated. As you move along, you see, for example, frequencies of A, C, Gs, and Ts differ. At some point, you want to say, oh, I'm probably in a coding region, so I should move over and use the other transition probabilities. Okay. That's just one example where these can get fairly sophisticated to better model something that you already know and then deploy that on something that you don't know to try to make better predictions about the probability of the hidden words, basically. Right. So many, like what we're doing here with finding hidden motifs or hidden words, has applications in lots of genome analysis, right? Whether it's trying to predict genes or trying to predict transcription factor binding sites, trying to predict folding patterns just based on DNA sequence, or we haven't really talked about amino acid sequence. The folding pattern really is more often used for amino acids than for DNA folding, but still equally applicable. It's just the reason we do DNA for the most part when I'm introducing this is that if you expand this to the amino acid sequence, and right now we have a much larger table, right? Because we have a potentially 20-something by 20-something table of amino acid transitions from one to another. Okay, so back to this issue we had with in the Vibrio origin of replication, we've got some patterns. We've got multiple patterns that happen 3 times. And so the question is, what's the likelihood that we would see that just by chance, right? Just random sequences of nucleotides. They don't have any biological meaning. Well, if you look at the multinomial, right, that first model that I talked about, what's the probability of seeing any particular 9-mer? Well, it's 0.25, right? That's the frequency of all the nucleotides raised to the 9th power, right? Because it's whatever 9 nucleotides you have in a row, doesn't matter their actual identities because they all have the same frequency, right? So that's the probability of getting any particular sequence. And then we have 500 base pairs in which to look for that. So if you multiply the probability of seeing it in 500 base pairs, you get 0.02. That's how many times you would expect to see a particular 9-mer just by chance. So seeing it 3 times is actually not super likely. And yeah, this notation is just telling you it's not, that's the probability of seeing it once, and the probability of seeing it 3 times is 0.002. I mean, I did not explain that very well, right? 0.25 to the 9th, that's the probability of a given 9mer, and then you've got 500 base pairs to look in. That gives you the probability of seeing it 1 time as 0.002. And we've seen it 3 times, so the probability is that probability of seeing it 1 time raised to the 3rd power. Okay, so it gets really small. Very unlikely. Okay. So, but basically, I'm not sure why the slide had that hidden, but if you're looking through, that's the question the bottom thing is answering. So this is expanding 0.002 to the 3rd, right? That's 8 times 10 to the minus 6. If you look across the whole Vibrio genome, you would, which is 4 megabytes, 4 megabase pairs, sorry, you would expect to see any random 9mer 32 times just by chance. And if you extrapolate that to a human-sized genome, you would expect to see that any random 9mer 24,000 times purely by chance, right? So you can actually get some frequent words if you're looking across, having to look across a whole genome. You know, a random 9mer in the human genome will occur 24,000 times. So, you know, I'm just pointing that out to say just counting frequent words isn't enough unless you have some model for how many times you expect it to happen by chance. And again, this is the most naive model, right? Where any 9mer, because we did 0.25 for all nucleotides, it has the same probability. But if you make more realistic models, then some 9mers would become more likely than others. So if we now focus back on our cholera genome, Vibrio cholera genome, one thing to notice here is that it's not just that there are 3 of them, which is pretty unlikely in a 500 base pair sequence. The ones that I colored green and red, are actually reverse complements of each other. And DNA A is not strand specific, so actually something going on where either there's a conformation where these things are together, or there's actually 6 potential binding sites here. It's really the second one. Okay. It is super surprising to find a 9mer that occurs 6 or more times within a 500 base pair window. So what if we apply our hidden word problem to this other Thermatoga petrophila? Well, unfortunately, we get no occurrences of the Vibrio cholera one. So if we just looked, right, we just took the very first Roslin problem and looked for how many, count that word or count either one of those words, you get 0 matches. But when you apply our frequent words problem to it, now there's actually 6 matches. So we've got a similar problem to what we had originally in the Vibrio genome. And that's just because the DNA transcription factor recognizes a slightly different binding motif. So one thing to recognize, 2 of these happen to be reverse complements of each other, which is cool and helps out. But even then, going back to our original question, can we call that the hidden message? Is it unlikely? Well, we already know that seeing this 5 times, right? There are 1, 2, 3, 4, 5. That's super unlikely. And so it has a high probability of being a binding site for DNA A in Thermatoga. But, right, if we go back to this problem, oops, not that one, right? If we notice back here, what if we weren't given just the 500 base pair window of the origin of replication? What if we had to actually look through the whole organism's genome? Well, now, I don't know if, right, this is actually not a hidden word by those definitions, because we would, I don't remember exactly the size of Thermatoga's genome, but let's say it's roughly the size of Vibrio, so 4 megabases, we would expect to find 24 hidden messages just by chance. So how can we find the origin of replication still, right? That's the big picture question that we're looking for. Even within the 500 base pairs, the frequent word is defined by, 3 in Vibrio, and now we've got 6 in Vibrio, 5 in Thermatoga. So we somehow need to be able to find sections of the genome where these things are clumped together, and clumped is going to be the key here. So up to this point, we've been given the region of the genome where we need to look for the origin of replication. But now the next step is, how do we find that region where we need to look? So how do we find the origin of replication first, and then find the hidden message within that origin of replication? Okay. So we've got that 500 base pair window, and we can count k-mers and find hidden words, no problem. What happens if it's not known? So this is basically what we need to do. We need to slide along the genome, and for each section of the genome, count the frequent words. So now, not just enough to run our frequent words algorithm, we need to slide across windows of the genome and count frequent words. So let's define what a clump is technically, right? So k-mers form an LT clump inside the genome if there's a short sequence of length L interval of the genome in which it appears many times in at least T. So for number 16, finding patterns forming clumps, we won't have to deploy the multinomial or a Markov model. So we're not going to actually have to program those, because we're going to be given the length. So how big a chunk. So far, we've looked at 500 base pair sections of Vibrio and Thermatoga, but that window size could be anything, right? We don't necessarily want to constrain ourselves to saying the origin of replication has to be 500 base pairs. So that length is going to be L, and Rosalind is going to give us that number. And then how many times does it need to occur, right? Which is where you would use a Markov or a multinomial model, is actually also going to be given to us. We don't have to use those. It's going to be given as t. So we want it to happen at least t times. And one way to think about this, right? We have so far been using max count and just returning whatever the maximum was. Now t is effectively the maximum. It's telling us that we need to keep everything that has occurs more than t times. Okay. And then just the translation of that formula, right? A clump is a genomic window where the k-mer appears more times than predicted by chance. And that predicted by chance is given as t. So if you go to finding patterns, yeah, that's okay. So also taking the next step here in terms of your development of coding and algorithms is I haven't given you pseudocode. I think they have pseudocode in the chapter. I didn't go back and look before I started the lecture. But there's nothing new that you need to know coding wise or even strategy wise, because you already know how to do sliding windows. So, right, if you think about before you just sit down and start trying to code, what do I need to do? In this case, I have to count k-mers that are 5 base pairs long, right? My patterns are 5 base pairs. The origin of replication size, L, right? So instead of being 500 base pairs, in this example, it's only 75 base pairs. And then to define a clump, we have to have a pattern that occurs 4 or more times. Okay. So, for example, we're going to slide along and get the first 75 base pairs. And then we're going to run our frequent words algorithm on that 75 base pairs for a k-mer size of 5. And we're going to keep any of the k-mers that happen at least 4 times. Okay. Then we're going to slide over one, get the next 75 base pair window, and do it again. So, right, run our frequent words, keep all the words that happen at least 4 times. So this is really the only new thing here is writing an extra layer of sliding window where we get 75 base pairs and then do our frequent words thing. Slide along, find frequent words, slide along, find frequent words, and so on. Okay. And then at the end, right, you have to maintain some kind of master list that is collecting all the patterns that met that criteria of happening 4 times within a 75 base pair window. All right, that's it for today. Keep on the lookout. I will post an announcement because I'm going to make up to number 16 due by next Tuesday. Okay. All right, happy coding.